Gracias. Bueno, continuando con nuestras charlas plenarias, tenemos la presencia del doctor Goldie, de quien también me permitiré presentar su semblanza. El doctor Goldie es profesor titular en el Centro de Investigación y Desarrollo Tecnológico en Electroquímica, CIDETEQ. Él es miembro del prestigioso Sistema Nacional de Investigadores de México, nivel 2, desde el año 2015. Tiene doctorado en bionanotecnología por la Universidad de Bombay, en India. Es autor de más de 100 publicaciones científicas, entre artículos indexados en JCR, capítulos de libros como el de bionanotecnología, conceptos y aplicaciones, y el de nanoquímica, síntesis, caracterización y aplicaciones. Ha participado también en ponencias en congresos. El doctor Goldie estableció un laboratorio de fotosíntesis artificial y diseño y la fabricación completa del laboratorio órgano on a chip aquí en el CIDETEQ. El doctor es especialista en nanomedicina, inmunoterapia, biosensores, órgano on a chip y fotosíntesis artificial. Se desempeñó como revisor invitado para más de 24 revistas JCR, dentro de las que destacan editoriales como Springer y Nature. El doctor Goldie ha participado en diversos foros de comunicación, incluidos periódicos, radio y televisión, sobre temas científicos propios de su área de desempeño. Tiene colaboraciones con diferentes universidades en todo el mundo, incluidas la Universidad de Texas, el MIT, el Instituto Italiano de Tecnología, la Universidad de Oxford, entre otras. Damos la bienvenida al doctor Goldie.
Muchas gracias. Muchas gracias, Alejandra. I hope I can take this session in English because I don't speak very good Spanish like like all of you. The first and the most important thing which I've been told to present here is talking about what are the implications of impedance spectroscopy in the area of detection of cancer, as well as we are talking with respect to the two-dimensional as well as three-dimensional systems or platforms, wherein we would like to work or comprehend how the cancer cells can be detected in an organ on a chip model, because impedance spectroscopy is considered to be a very important technique so that we can understand how these cells behave under the influence of electrical conduction or electrical impulses. So these are the reasons, and then what we are interested in is like we would like to understand that how the cells undergo adhesion, cell migration, invasion, proliferation, viability, and induce apoptosis within itself. So these are the complete overview of the cells which we would like to understand, and organ on a chip is a model. So why we are interested in having this system of organ on a chip? What is it? Because organ on a chip is a model or a platform wherein we can have a controlled environment or a completely controlled microenvironment wherein we can regulate or control the cells in a particular channel. You know, if you are working in a system which is talking about mice, or when you're working on a system in a very complex microenvironment, then it becomes very difficult for us to understand how the migration of the cells happen. What are the molecular mechanisms of migration existing within it? What are the invasion molecules? What are the invadatory molecules which are responsible for the cancer cells to move towards a particular direction? Or what are the mechanisms how immune cells they traverse all the way from one particular region, for example in blood, because you will find that all the cells, all the immune cells will be either in in the spleen, lymph node, or it will be in the circulatory system. So we would like to understand when and how these cells, these immune cells, they get activated. Because most of the time in cancer, what happens is there is a problem in the immune system, which it's difficult for them to detect such cancer cells, especially the macrophages. Macrophages are certain cells of immune system which most of the time becomes worse. There are good macrophages as well as bad. So we would like to understand how these macrophages, they play an important role also for the detection of cancer cells, and then they motivate the cells, they motivate certain apoptotic inducing mechanisms so that the cancer cells will die at the helm of these interactions between the immune as well as the cancer cell. So all these mechanisms, you will imagine that it's very difficult when you're dealing with actually a human body. It's really very complex system. So we need to simplify this system, but equally we would like to mimic them in terms of the body as well. Okay, so now we would like to move ahead with the first slide here. Most of the people are aware of this kind of physiology, human physiology, wherein we are talking about human physiology in terms of senses, endocrine system, digestive system, reproductive system. These are the Eastern type of physiologies which people have been mentioning. And then there is a concept which is this is the Western type of technology, wherein we are Western type of explaining physiology, wherein we are talking about the lymphatic system as well as the circulatory system. So the whole body is connected via two systems, the most important systems, that is lymphatic as well as the circulatory system, the blood. So this is the difference between the two systems, and these are the reasons, and all these lymphatic and these systems are responsible for taking care of all these overall physiology of the body. If these systems doesn't exist, then these organs will not also exist. So that's the reason why these systems, the circulatory and tegumentary, as well as these lymphatic systems, are very critical in maintaining the balance between these organs in the body. Let's talk about the cell culturing. Now the thing is, we are all aware of, when we talk about physiologies, all those tissues, all those organs which are converted into tissues and then later into organs, into the cells. So those cells were grown previously in different kinds of systems. And the most important system for the growth of the cell culture system was a two-dimensional system, wherein we had this rocks which was responsible in the year 1885 to grow the cells for the first time. And then this whole sequence of events happened, and then we are all quite aware of a culture medium which is used for the cell culturing, and that is known as DMEM, Delbecque's minimal and Eagle's medium. So this particular medium is being used even now for the growth of the cells. All right. So this is the sequence of events which have happened for the growth of the 2D systems. Then, now, what's the difference between 2D as well as why we are discussing about organ on a chip? Because organ on a chip is something which is dealing with three-dimensional systems. And what do you mean by three-dimensional systems? I hope everybody is aware of that in the body, we have cells and they are all three-dimensional. Am I right? We have a three-dimensional system within the body, right? And when you talk about a two-dimensional model like this, is this kind of two-dimensional model, what happens in this kind of model is the cells will grow, and as the cells are growing, and there will be a confluency, that means there will be one single monolayer of the cells which will be growing. And as soon as it reaches to that confluency, it will stop growing. This never happens. Of course, there is a contact inhibition concept inside the body, of course, with the normal cells. But this is not the situation in the case of cancer cells. In the case of cancer cells, they will grow in all dimensions, and they have lost this contact inhibition concept, and then they will be three-dimensionally, they will be growing a lot. But we had the capacity only till now, I mean to say 5 years back or 10 years back maybe, we had only the capacity of growing this kind of setup that is only one single monolayer. Now what happened with this? When you're talking about single monolayer, you might be knowing that the surface area is enormous. So for example, if a patient, if a medical doctor, he wants to give a medicine to the patient, what he will do, he might tell to get a biopsy of the patient, and then he will recommend to the laboratory guys that, okay, grow these cells into your system, and the lab guy will be having only two-dimensional models. What will happen? He gives a doxorubicin drug into the two-dimensional system. It works. Effectively, it works. And he says, hey, there is a 90% killing of the cells. You can easily go with this doxorubicin and then enjoy. The patient will be living. But to my surprise, in 3 months, the patient dies. Okay, why? Because the patient was suffering from triple-negative breast cancer, and this kind of triple-negative breast cancer is very, very aggressive type of cancer, and disease, I mean to say, and moreover, the patient who was the cells were treated in the two-dimensional model like this, in this kind of setup. Due to the high surface area, all the doxorubicin was absorbed in this particular system, but the same doxorubicin was not absorbed by the cancer cells inside the system, inside the body. It was very less absorption happening. The uptake was very less. So with this idea, it was confirmed that we cannot be dependent on two-dimensional systems. Further, people were working with mice, fine. If they're working with mice, what happens is, with the mice, they are not exactly mimicking the human body. So that was also another situation for us. People started injecting, of course, even now the same thing happens. They will take the samples, the breast cancer samples, inject into the mice, and they will grow the tumor inside the mice, and then they will start doing the treatments, and they will get a hell lot of results. And based on that, we could not treat cancer completely. Of course, the patient were not treated with that also. So nothing works in that situation. Then people have come up with this concept of 3D system. So why 3D? Three-dimensional models. Why? What's the reason behind that? The 3D model systems are basically, they have tunable, relatively low stiffness, and then they are basically having all the matrix porosities and the motilities and all the kinds of gradients which are required and all the kinds of factors which are responsible for their growth, and that too in a three-dimensional way that has been seen to it. And you can see here, I think I have this. So you can find here that these are the three-dimensional models which are being growing, and they are considered to be spheroids or organoids. Now, spheroids or organoids, what are these structures? These are complex clones, you can say. It's a complex clones of cells, but it's a mixture of clones of cells wherein it will be, again, this situation is like a unregulated tumor, kind of, or unregulated complex of cells which are being growing inside this particular environment, wherein all the cells, the immune cells, the epithelial cells, the mesenchymal cells, the cancerous cells, the tumor cells, all of them are mixed into this particular setup, and they are all growing in a particular fashion. But again, the situation is unregulated fashion. Though it's three-dimensional, but again, it's growing in a very, very unregulated fashion. We don't know where are the tumor cells existing, how the cytokine mechanisms move, how the migration of the cells happen. We never know any of these aspects. So, but then this is something which we have achieved till now. This is something which is very important. Even this is very critical. So then people started working on this kind of 3D dimensional systems, wherein, they have developed a kind of different kinds of organoids. Now, this was a successful achievement. It was an achievement for the scientific community, wherein they were absolutely having all the cells coming up to form a tissue. All right, and then there are different kinds of tissues which were made using stem cells, pluripotent stem cells, hematopoietic stem cells. All those stem cells were used, and then after that you had different kinds of organoids like intestinal and gastroloids and hippocampal, kidney, liver. All those different kinds of organoids were formed. Okay, it was a great achievement because it is mimicking. It is mimicking the system. It is mimicking the tissues inside. But yes, but the problem is that the biggest issue is that even though it's mimicking, the system is mimicking, but we could not actually understand what's happening between the cells. That was, again, I will constantly pose this question again and again during whole of my session. And why I'm posing this question, you'll understand as and when we are moving ahead towards this presentation. Fine. So what we did, and there is a group, Dr. Alejandro Centella, he's from UNAM, Biomedicina. So this group is receiving samples from the patients. So what they did was they tried to have a comparative studies between a two-dimensional structure. This is a two-dimensional model, cellulose and monocappa. This is the one, and this is all triple negative, it's a triple negative breast cancer cell lines. And here they had, and this is a spheroids. It's a complex. It's a three-dimensional structure. So what's the difference? In this three-dimensional structure, he could confirm that which particular drug needs to be given. All right, he could confirm with this that, okay, these are the carboplatins and doxorubicin, and even after that, he could also find out that what are the concentrations which are required for these drugs to be delivered at this particular point. So this was confirmed using this spheroid structures, and then the doctors were, and these are basically from the patient sample directly from the hospital. And this confirmed that this spheroid concept works. And finally, the carboplatin and doxorubicin, these are the two drugs which are very critical for the killing of the cells. All right, fine. So with this, the doctors were given the information that these are the two drugs you should give to your patient. And yes, the patient is surviving for months and years together. So there is no problem in this situation. And of course, but problem is that the question still remains the same. I don't know, even anybody does, like people don't know how the cell migration takes place, how cell adhesion takes place. What are the factors, the growth factors responsible, or the stromal factors which are responsible for the interaction of the cells onto a substrate? And once the interaction happens, and how these cells, even if they want to move out and they want to migrate, for example, metastasis, people are aware of, like people still, we don't know what are the actual mechanisms of benign cancer and the metastatic cancer. What's the difference between the two? And that is the biggest question arriving amongst all of us. And now then the people, the scientific community started understanding that we need to have a three-dimensional model. Of course, that's true. We need to have a three-dimensional model, but we need to have a complete, a separation of the cells. That means immune cells must be there. Okay, now you can see here this kind of setup. Yeah, this one. You can see here, these are the organs. For example, lung. Now, lung is encapsulated by circulatory system as well as lymphatic system. And now this lung, which is encapsulated by both the kind of setup, what will happen after that is that the immune cells or the blood cells, they will get into the immune cells will get into the tissues which have been infected. So basically there are three different setups here. That means we are talking about the organs, We are talking about the endothelial cells, and these endothelial cells are a part of the blood vascular system. And we are talking about the immune cells, which are a part of lymphatic system. So if we make a setup, or a microfluidic setup, or a organ on a chip setup, wherein we have all these three platforms placed at a time in the same position, all right, but equally they will be separated also. So what will happen? And we are mimicking exactly what's there in the body. You can imagine, you can see here that this particular way, these are the arteries, veins, all of them, they are like moving, like they are like a pipe. All right, they are all completely a microfluidic system, basically, which are completely separated, but they meet each other only via liquid or some gases. So there is exchange of liquid or gases happening, but then also they are separate entities. So basically what we are interested in doing here is this kind of ideas here. You can see here. So what are we planning? What are we interested in doing? That this is a kind of a setup, all right, and wherein we can have in one channel we can have a kind of the immune cell. Here we can have the endothelial cell, that means the blood cells, and here we can have the cancer cells. So all these three things are placed in a single setup, in a single microfluidic channel, but equally they have been separated from each one of them. So this is what we would like to do. And once we do this, we will understand that yes, all the cells, they have an important role, and also we can understand that which kind of cytokines are being secreted by which kind of cell. For example, there is a cancer cell, and this cancer cell will be secreting, certain molecules, cytokines or chemokines or whatever. Then they will traverse through this particular setup, that means endothelial system, and then they will traverse through the immune cells. And then this immune cell gets a signal that, yes, I have been receiving the cytokines regularly, continuously. Now I have to traverse all the way from this particular region, from this point, directly into the place where the cancer cells exist. So in this way, we are understanding what is the mechanism of activation of the immune cells. What is the mechanism of migration of the immune cells, and what is the mechanism of invasion of the immune cells? So these are the concepts which we will understand only when we are dealing with a regulated fashion of the system. Yeah. Then, when we talk about microfluidic setup, we have all the cells spatially controlling, spatial and temporal controls are there, and we will understand what's going on in the whole setup. And then there is a complete discrimination or a differentiation of the setup, perfectly aligned. And you can also have a complete automated PDMS based microfluidic setup, wherein this is a cell culture system. They have been integrated with the micro sensors, and these micro sensors will be detecting glucose and pH and all other lactates with this. And then we can have and even we can also detect the apoptosis induction in the cancer cells, and that is basically done by the electrochemical impedance spectroscopy. Fine, this is talking about the applications. All right, so what we have done till now, I'll just explain you what we have done also is we have identified that there are transferrin receptors existing on the surface of the lung cancer. We have done using this proteomic analysis, and based on the proteomic analysis, we could identify that yes, these are the receptors which are existing enormously on the surface of H69 cells, which are considered to be lung cancer cells. And then we could identify that what are the roles of these transferrin receptors, how they work against how they are activating different mechanisms so that the lung cancer cells can proliferate innumerable times. Then we also could detect this using flow cytometry, and we could also do immunofluorescence technique, wherein we could find that all these receptors, all this on the plasma membrane, these receptors are enormously present. They are like approximately 40 times higher in comparison to a normal lung cell. So this transferrin receptors were a success for us, wherein we could use this transferrin receptors integrated with the nanoparticles, and then after that we induced hypothermia, and then we tried to kill these types of cancer cells. So this was one kind of work which we have performed. Then we also identified, using machine learning and artificial intelligence, we could identify what are the different markers present on the surface of this triple negative breast cancer. So we could find that the most important marker which is existing, we could analyze approximately 38 genes, and we could find that there is one marker which is considered to be CD44 receptor, which exists almost 30 times higher. The numbers are 30 times higher. That is, overexpression of this CD44 receptors exist. And we could identify using flow cytometry that these are the markers. We could confirm. This was identified during artificial intelligence or machine learning work. And we could confirm it when we analyze the samples, or MD 231 samples, and we could confirm using flow cytometry that these are the markers which are existing enormously. They are overexpressed in the samples. And then, of course, we have used those markers again. We have exploited this marker again for adding docetaxel with the nanoparticles and injected into the cancer cell, which is having the triple negative breast cancer, and induced again hypothermia. And then we had done the killing of the cancer cells, especially triple negative, which was an aggressive type of cancer. You can imagine this particular patient dies within 3 months. It's highly aggressive thing, and there are very less treatment strategies for triple negative breast cancer. So for that, we had initiated the work of hypothermia, of course, number one, as well as this kind of CD44 directed therapy. Okay, this was one of the therapies. And the third most important thing for triple negative was we could identify a molecule, B-calutamide, which is being given to a prostate cancer patient. And we could find that the same drug can also be given to the patient of triple negative and breast cancer, and the patient's life increases to many number of years. So that is the kind of work which we could do. Then after that, why this impedance spectroscopy is useful. But before that, you should also understand that we are interested in identifying that, okay, how the cells will. This is the 2D transfer system. And in this 2D transfer system, we are interested in identifying that, okay, there are macrophages on the top, immune cells, and at the bottom, there are cancer cells. And if you treat this setup with a drug, then these macrophages, they will traverse through it, all right, through this porous membrane, and then they will go towards this cancer region. So this is the actual mechanism of 2D transfer system. And the same thing we are doing in a microfluidic setup, wherein we would like to check the same mechanism and how these macrophages, they traverse through the porous membrane. This mechanism we are trying to understand. Okay, and yes, now let's talk about the electrochemical cell substrate impedance spectroscopy. Now, why we are interested in doing this kind of work? The reason is, okay, first of all, I think we would like to understand, and everybody knows about it, there is a glass, chromium, then gold deposition, and then after that, we have most of the time this UV pattern photoresist, and then we have this electrode and trace being generated. These are the electrode formation mechanism. And then what we are interested is we are having a cell adhesion strategy. Now, what is this cell adhesion strategy using this electrochemical impedance? How it works? How is it so effective for us? Because you can imagine that whenever the cells are being attached onto the electrode, all right, so onto the electrode of these gold electrodes, once they are attached onto the surface, then immediately the impedance increases. And as soon as, these cells are growing, okay, as the cells are growing, they're completely occupying the electrodes. And as the electrodes are being completely occupied by these cells, the impedance level further rises. And as it increases further, and once these cells are about to die, there is apoptosis or any aspect of induction of apoptosis happens, or paraptosis, or any other kind of apoptosis works here, necrosis also, then suddenly there will be a drop in the impedance. And based on that, we can identify that how the cell adhesion happens in this particular mechanism. We could understand using electrochemical impedance spectroscopy, we could understand how the cells are growing, and more often how adhesion happens. Further, we can also understand how migration happens, how proliferation happens, and how apoptosis. So we can get the signals for each one of them. All right. Okay. And with this particular graph, you can easily identify that actually what is the state of the cell. Okay, how the cells are behaving inside the system. So the same. So I just want to create a background here again. That we have again we can have a 3D electric cell matrigel substrate. Okay. So what is happening here is, again, there are the gold electrodes, and then these gold electrodes will be having the medium and with the cells adhered onto it. And once the adherence happens, then we can easily identify or detect how the cells, and this happens at a particular frequency. Yes. So now the frequency is approximately what generally people have used here is 0.5 to 10 kilohertz. That is the frequencies people have used. Now, this is a very important thing wherein it exploits flow cytometric analysis. Now, people are aware of flow cytometry. What happens is flow cytometry is responsible for cell sorting. We can sort the cells. So here there is a concept of impedance flow cytometry, and there is electrochemical impedance spectroscopy. So this flow cytometry is responsible for sorting the cells. And then these sorted cells will be getting into the EIS electrodes. And then once they get into the EIS electrodes, then the actual state of the cell will be considered, will be understood. And once these actual state of the cells are being understood, then accordingly, we can identify what is the state at what particular time, and how can it be detected. So basically, we can understand the proliferation of the cells happening, how it happens. So the complete proliferation strategies can be understood using this EIS technology. Then I think I can. Okay, so this is the kind of final model which we are developing here in CIDETEQ. The thing is, we are having, as I mentioned, that there are like three channel systems wherein we have, as you all know, that in one channel we can have the cancer cell. Maybe it can be anything. One of them will have the cancer cells. In between there will be the endothelial cells, and in the last channel there will be immune cells. And these are the electrochemical, interdigitated electrodes which are connected, and they are responsible for the detection of the cells, okay, and detection of whether the cells are proliferating or they are not proliferating. What is the behavior of the cells? And moreover, you can see here there are minor micro channels here existing in this point, wherein they will be responsible for the movement of the molecules, not the cells. All right, they will be just responsible for the movement of molecules like chemokines or cytokines and many other systems, so that the markers we can easily identify them, and we can also detect whether the cancer cells are being killed or there is any apoptosis induction within them. So with this, I thank you all to give a small to listen to me for towards this complete understanding of the perspectives of organ ownership with respect to this impedance spectroscopy. I'm very thankful to all. Yeah, if anybody's having any question, of course.
Muchas gracias, doctor Goldie. Tenemos unos minutos para un par de preguntas para el doctor.
¿Pueden ser en español, Goldie?
Sí.
Sí. Con toda libertad. Él entiende el español, solo que la plática era un poco especializada. Poco. ¿Quién guste preguntar?
Thank you very much for the interesting talking, Doctor. It is very valuable, all the concept that you have presented to us. Particularly, it results from my personal interest, the proposal of this measurement about the impedance spectroscopy in the 3D culture. Have you considered in your further experiments about the quality of the control? I mean, in the classical 2D cell culture, we place as control an exact number of cells.
Yeah.
But in this case, this number will impact in the charge behavior directly. So could you give us a little bit more insights about that?
Yeah. You're talking about the number of the cells which will be placed inside, right? So the thing is, this impedance spectroscopy also helps us in understanding the viability of the cells and determining the number of cells present. Because most of the time we will be adding approximately 10⁻² or 10⁵ cells per ml. Now these cells, as soon as, that is a basic idea, that as soon as the cells are interacting with these microelectrodes or interdigitated electrodes, they will immediately give us a signal that what are the numbers of the cells which are existing. And further, they will also tell me, because, okay, in two-dimensional models, people are already using in two-dimensional models with a 12-well or a 24-well plate. And these 24-well plate are placed in electrochemical impedance counter. And they will be actually telling the numbers of the cells which are actively growing. And they will also differentiate between active growing and apoptotic induced cells. So we can get a perfect number using this spectroscopy for sure. And that's the reason the word comes electrochemical cell substrate impedance spectroscopy. So that will be quite useful technique in this case, because most of the time, as you have mentioned, okay, why we have come up with this idea of introducing this electrochemical spectroscopy. That is the idea. That is a motto behind that. That's a perfect question you have asked. When we are not placing these EIS technology, it's difficult for us to count the numbers. We'll simply add, and we never know what's happening. It's a black box for us. And when we have this electrochemical impedance technique, then it's not a white black box for us anymore. We will identify what are the numbers. We'll find what is the logarithmic phase also in that case. What is the logarithmic phase of the situation of the cells, whether they're growing, they're getting killed, and if at all we want to put any nanoparticles or any drugs into it, we'll also understand, okay, what are the numbers of cells which are living or dead, all those aspects. So that's the reason this EIS has been introduced into this technique, into this organometry. Yeah.
All right. Thank you very much, Doctor.
Thank you, sir. Thank you.
Le cedo el micrófono al doctor.
Thank you.
Thank you.
I will to ask in Spanish.
Está bien, está bien.
Okay. Gracias por la ponencia, muy interesante. Bueno, me gustaría conocer si hay algo en relación a la detección del cambio del fenotipo, el fenotipo tumoral de cáncer de mama al tipo mesenquimal epitelial, que es lo que se ha estudiado que da lugar a la metástasis.
Sí.
No sé si haya alguna investigación en donde a través de impedancia, de espectroscopía por impedancia, se pudiera detectar este cambio del fenotipo celular en las células epiteliales a mesénquimo epiteliales.
Correcto, sí, muchas gracias para esta pregunta. De cosa es que normalmente tiene algunas células de cáncer y tiene interacción con electrodos, y donde tiene todos los electrodos de oro que tiene una interacción con nanopartículas de oro también y con algún tipo de anticuerpos, y tiene una integración con células de cáncer. Y en esta situación mucha gente tiene conocimientos, y también de grupo hay algunos artículos que mencionan que hay incrementos de matrix metaloproteinases, de concentración de matrix metaloproteinases, y después tiene una interacción de electrodos, y después tiene una diferente behavior de impedancia cuando tiene, por ejemplo, de células de cáncer tiene un crecimiento en células de cáncer. Y también cuando tiene cambios de morfología también tiene un cambio de impedancia en esta situación, porque usted aquí tiene mediciones de dielectric measurements. So aquí usted tiene mediciones de dielectric, cambio poco de morfología, cambio mucho de impedancia. So esa es una buena relación también en este caso. So es buena cosa para integrar espectroscopia para identificar de morfología, y también algunas veces que tiene, por ejemplo, cuando tiene metástasis, en metástasis de células no tiene tanto adherencia para algún electrodo. So cuando ellos quieren ir y después inmediatamente tiene cambios de impedancia y puede conocer. So impedancia es muy buena técnica que tiene una discriminación de células de cáncer y también células de normal. Y también esta buena técnica para detección que, es benign y también es metastático. So, esta cosa también puede discriminar bien con esta técnica de impedancia. Por eso, sí. Gracias. Muchas gracias. Sí.
Alcanzamos a otra pregunta. Un momento.
Sí.
Is it possible to correlate the cytometry with bioimpedance with the regular cytometry?
Correlate cytometry with impedance?
Yeah.
You're talking about flow cytometry, right? Yeah. Yes, because if you understand what is flow cytometry, wherein there are two things which are going on in flow cytometry. One is sorting of the cell, that is separation of the cells. All right. And the second thing is they will be detecting the cell, but they are all in liquid form in cytometry. So they liquid form, I mean to say that the cells are always in a solution. That's the biggest problem in flow cytometry. We are not detecting any cell which are adhered. Okay. And when we are incapable of detecting adherence of the cells, then we are incapable of understanding how metastasis takes place. So this technique that is, which is also known as, impedance flow cytometric analysis, wherein we can join the two, that is flow cytometry as well as impedance, they can be joined together, and then we can identify how the cells behave when they are interacting and how the cells behave as soon as they're about to leave from the electrode. So this complete mechanism or the process can be studied using the impedance, the signals, and that is what I think which is required. There is a correlation between the two, and the best thing is we can understand on the surface adhered cells, but in flow cytometry with the conventional technique, we cannot do any of those kind of detections when it is in adhered onto anything. It should be in the solution form. So this is an added advantage to this flow cytometric analysis. Yeah.
Muchísimas gracias, doctor.
Gracias.
A nombre de CIDETEQ, así como del comité organizador del 3er Coloquio Mexicano de Bioimpedancia, emitimos una constancia agradeciendo su participación, así como un obsequio.
Muchas gracias, doctor. Muchas gracias. Gracias.
Con esta parte damos por terminadas las plenarias que teníamos programadas para este 3er Coloquio Mexicano de Bioimpedancia. Les agradecemos a quienes están conectados en Facebook por su participación, por andar pendiente y por el interés en estos temas del grupo. Y con esto terminamos la transmisión por Facebook Live. Ahora